2019. And okay, my PhD topic is, I put one introduce slide after this one. I'll introduce my details after this title slide. So I'm more interested in DNA and eDNA based method to understand the biodiversity and to understand detailed reveal the information from biodiversity. So I also put some information about myself. So in last 4 years I have achieved to do something, and I have achieved to develop some skills related to my field. My principal interest in environmental DNA, as well as I'm interested in plant-animal interaction, metabiology, and population genetic studies. In addition to this one, I'm especially interested in wildlife biology and also in other ecology activities we work. So for me, it was very great experience to study here 4 years to understand my passion in this biodiversity field and to develop some skills. So can I start from here, my presentation? So I'd like to start with the big picture of art. As I was motivated by Professor Christy Daniel, who used to show this picture from NASA. This is very this picture is a biggest picture of the art, where we are seeing that the biotic abiotic factors are interplaying together. So this biggest picture of the art, and I would like to say that how much we know till now, maybe less than 15%, how much we know about our art, how much we know about our interaction. From here, with this question, I'm going to start with the details about the biodiversity. So I'm always passionate about biodiversity, to understand the details about what's going on in the environment, and to understand the details of the biological factors that are affecting our diversity. So we have two terms, eco and ecos. If we tell eco as with all the plants and animal and human living together in the era of anthropocene. But unfortunately, last few decades we have seen a huge decrease, I'll say huge decline of biodiversity due to human activity. So this is the recent report where we have seen that 69% of the world biodiversity already declined, and many species are going extinct, and some of them became vulnerable, and other stages near threatened. We are as far we know that this biodiversity in turn has some other effect on human beings also. So by clarifying this report, I would like to show you the highest decline rate was in Latin America and Caribbean is 94%. And in addition to this, the Africa, we have strong biodiversity. We also have 66% biodiversity decline. Together with Asia, we have 55% and other places. So what is the factor is affecting this biodiversity decline? We need to understand this one. In the big picture, we can say that the climate change and increasing Earth's temperature is affecting the decreasing of biodiversity. Here I put one example, that's where increasing of Earth temperature and the decreasing of biodiversity. You can see that from the era of Anthropocene, the 4% Earth temperature increase, the biodiversity decline up to 75%. If we go more details to understand why this is happening, we can figure out 5 main reasons behind this decline. One of the main reasons is the land use. We are generally grabbing our earth, making out the lands to use by ourselves, agriculture activity, we have deforestation. Those activities are affecting the habitat of local wild animals. Next is the pollution. Pollution is also another key factor for decline in biodiversity. Another one is the species overexploration. For example, if we continue to eat fish this way without knowing their actual biology, we will lose half of the population of our fish by 2050. Another one is climate change, as I focused before, and the other one is invasive species. All of you maybe wonder why I put invasive species there. Is invasive species too much affecting in biodiversity decline? Is there in a parallel relation? Because how much invasive species can affect in biodiversity? Is it a huge effect? Yeah, I'm going to focus on the invasive species now to show that this is the equal relationship and equal effective threats for biodiversity with these things. So to prove this one, I'm going to show you recent data from the Triple Nodal in 2023. This paper summarizes all the literature to understand that which species, like which factor is causing more economical damage in the world. This is storm and other natural activities. And I'm very surprised to see this data. This is biological invasion, causing huge damage, economical damage to the world. And if we go to more details in the picture, here we can see that classification of loss in USD in different sectors, and where the biological invasion have a significant loss just after the storm. And in some cases, in some places also, they are over the storm and they cause huge effect. If we divide this one on the basis of continent, we can see that the Oceania and Pacific side, where the islanders just like reside, the island are very susceptible to biological invasion because they have a unique biodiversity and they have a unique species interaction there. So if invaded species occupy their space, there is a huge effect of the biological invasion. As well as our Asia, tropical Asia, here the effect of biological invasion is very high. The economic damage also so high. So invasive species are equally effective to cause the invasion and damage, in economical damage. But in which way they do this one? There is a 5 or 6 principle in our ecology. As we know that at the first they start predating their local plant and animals, I will say, and then they will start competing with the local native species, and then obviously another reason is the hybridization, and then they change the local species interaction and habitat alternation, and obviously spread of disease is very costful for the human beings. So in those factors, I'm going to focus on one of the most invasive species in the world, that's called golden apple snails. Those snails are they are very invasive to many cultures. They are very successful for invasion. Every invasive species is not successful, but they are very successful in invasion. Once they brought to the Asian country, they became a successful invader. But in the native country, they called as useless, and also the no reason behind that. I'll say that harmless and useless in the native country. But here they turn into serious destruction. So in the 1980s, Taiwan started to bring the golden apple snails. I'll say collectively as a golden apple snail because many species are brought together from South America, and we don't know which place of South America till now to Taiwan. And people have started doing for the aquarium trade and started as a food product and for their high protein rich. And later. But later they do not happy with that the texture of the snails, and the market soon dropped. And farmer was like they are not well known to how to do with the snails, so they released those snails in the outside of the field. And these snails are like highly propagated; they multiply very soon. And in the meantime, during the business, this snails was transported to other Asian countries. Soon after 1 year, 10 years, this became a serious threat into the Taiwan biodiversity and also the rice economic production. And red alert was conveyed at that time, and the government will take a national protection act, and then they try to remove those snails. But after few hours, those effect those national policy are stopped, and we are not taking any action on those snails, although they are present in the high amount in the field. So if we consider the global economy again about the gastropods, they have an aquatic and they have a terrestrial gastropods. Pomacea caniculata, the one species I'm telling about inside the golden apple snail, there are 4 species. So the Pomacea caniculata is a major responsible for 90% of the global economical loss. If we consider that in the global economy, there is a 3.94 billion loss happened from 1961. In Asia, the figure is like 90%, 3.71 billion loss due to the snails, and majority of responsible for caniculata. But I would like to tell you, the story just started here because this species is not originally identified properly here. There is a mixture of species living together and hybridizing together. We don't know. We do not have any idea that how many species are dealing with what kind of genetic structure they have, and how to deal with the species. So basically this background, my research question is like, are there multiple species of Pomacea living in the Taiwan? So we can take species action for those species. We need to know that how their distribution is. Then we need to know that these species are generally brought from South America or due to the founder attack, because other study also found that, like from China, Japan, they studied their origin and they found that this species directly come from the South America. But as the Taiwan was the center for the invasion, without knowledge of biological molecular data, we cannot say that which one is the founder effect, which one is the Brigitte effect. I mean, which one is from where the snail came into Taiwan first or other country first, and how this spread its actual path of invasion. So I need to understand this one. And then, as we say, Taiwan is the center of invasion of Pomacea throughout the Asia. We need to understand this one, because this is very a huge full effect in the founder effect and the Brigitte effect when, in case of population invasion. So we also need to understand that is that genetic diversity is a key factor for establishment of Pomacea in Taiwan. Why this species only established? Why other invasive species failed to establish? We need to understand this one. And this also, as Taiwan has a highly, their geography is like as an island, and also they have a separate partition of a different geography, like south, east, west. They have a separate temperature and like they have a separate environmental factor. I'll say a little bit. And then, is there any effect of geography of Taiwan with the snails distribution? And most importantly, are they hybridizing in Taiwan? There is very challenging effect that if they are hybridizing, they will put more strong backup to establish themselves. And yeah, we need to understand that these species are carrying out life-threatening disease of human angulosis. Is there any chances of parasite infection in those species? We need to understand in this point. And later, after understanding everything, we want to develop a technique that can detect the species very quickly and rapidly, so that we can monitor after, like if we start a mitigation or action on the government policy, we can monitor those species very quickly. So above all, we also want to add another point that we want to discuss, like we want to focus on this thing, that how about single species? We want to study the species interaction of with the environmental lineage with the other species, how they are affecting the native snails. Is their native snail population is declining based on the species? So let's, I'll start. We started sampling of all over the Taiwan, about 41 spot, and outside the 14 county. We collected the snails and don't know the proper identification of them. And then we also collected their eggs and then the snails from the different rice field and drainage all around Taiwan. So basically, I would like to say that in this factor, this is Pomacea canaliculata, this is Pomacea maculata, and this species is Pomacea scalaris. These two species was reported from Taiwan. This species was not reported from Taiwan. You can easily distinguish those species based on the shoulder. This have a little bit rounded shoulder and this angulate shoulder. But for this species, the differentiation is very highly difficult because they are cryptic in nature. So we found few diagnostic characters from these two species. Based on the pallial pigmentation, the Pomacea caniculata is unpigmented and this is pigmented species. The shoulder, this is reported to be round and this is like angulate. And the shell surface, one is smooth, another one is rough. And the shell thickness, one is thin, another one is thick. And the shell length, Pomacea maculata is a little bit more bigger than Pomacea canaliculata. This is the general factor we have seen from the literature. So we also studied those factors. We also studied the quantitative and qualitative morphological character to understand that is there really any differentiation between these two species. So as we studied the quantitative character from the Pomacea species, we found that there is a correlation between the shell length and height and weight, and there is a little bit correlation. But if we focus on the next thing, if we want to compare them, there are not any significant character and significant difference between their like our keys of taxonomy. Only we found that the Pomacea maculata is a little bit wider than Pomacea canaliculata, but this difference is very less. In field taxonomy, it's difficult to identify based on those characters. We studied the qualitative character, as I said that the pallial pigmentation and the shell surface and shell thickness and shoulder shape are key to the species. But here we found that none of the character are specifically detecting key to the species. They have like alternation of. We also found the rounded Pomacea maculata. We also found the rounded angulated Pomacea canaliculata. So there is an alternation of character. It's highly difficult to identify them based on the morphology. So we are thinking about how about the shell? What is inside the shell? The shell taxonomy is based on like Pomacea taxonomy is based on shell. So we want to study a little bit more details on the shell. So we went through SEM analysis to check that if there one is thick, another one is thin. Is there any difference between their internal structure? So then later we have seen that they are made up of ploslaminar layer and then the strong pillar and outer inner layer, and they are mostly like similar in composition, and it is very difficult to need a high taxonomic eye to differentiate them, and that will be very challenging for us. Then we went to understand that if there is chemical difference between those cells, if there is a chemical difference, then we can figure out. So we take some Pomacea maculata and canaliculata. How I identify it properly later I'll show. So we take the identified one and Pomacea maculata, and we went through some physiochemical analysis through Raman, FDR, SEM, and XRD to understand that if there is a chemical difference. Also, if they have like some bioavailable compound, we can use those compound as like in industry, so that we can use those invasive species in our way. So in case of XRD result, we also match their Miller indexes and the peak distribution and the composition. They are made up of like calcium carbonate, and they are like we have seen that their peak similarity is a little bit similar, but we have seen some of the very few differences, and which is very difficult to identify up to species based on that. But from here we can detect their chemical composition. And the next one we also detected with the XRD in the fingerprint region. We have checked their, this is the raw shell XRD. We have checked their chemical composition and how their differentiation to understand their fingerprint. Although they have the similar like bond size made up of calcium carbonate and the presence of CO bond, amide bond, everything is like little bit similar. And this is also supported by Raman, where they have a similar peak size and intensity and as well as similar peak position. And finally, we detected those the original chemical compositions through ETX that they are made up of calcium and calcium carbonate. And we also like from here we cannot differentiate those snails very easily, but we can suggest that those characters can be used in the industry to produce like biodiesel or biolateral calcium in heavy metal removal, or we can use this species as we have like a huge number of species, we need to kill them, so we can use those species. This is the point. So later come back to the original study from that side. We want to identify those species. We haven't like take any measure to identify till now. So we went through DNA barcoding assay. So as clearly as that we are trying to find out what universal primer and if there is a species specific primer. Fortunately, there is a species specific primer developed by Matsuda et al. In 2008, and we have also a LCO primer, the common animal primer that we have. We amplified those primer with the snail species. We have few control species like Pomacea scalaris. We also have. We have also some giant African snail in our lab and other control species to amplify with. So we have checked that this universal primer can amplify all the snails, but the species specific primer can amplify only our target snail. The higher base pair, this primer is the multiplex species where this primer specifically detect the Pomacea canaliculata, and this one is specifically detect the Pomacea maculata. So here we can differentiate them. But in case of giant African snail, there is no band. So we can say that this is very species specific. With this one, we studied all the snails. We collected over 300 snails from different places of Taiwan, and we tried to understand their distribution and their diversity. So we find out that the Pomacea canaliculata and maculata are a little bit heterogeneous in distribution. Their distribution is not totally equal, but they are sympatric. They are living together. The distribution of Pomacea canaliculata is throughout the Taiwan, but distribution of maculata generally restricted to the south part, and some of the represent we have found from here. But other places we could not detect in any sampling in the Pomacea maculata. This is very surprising. In whole the Asia, the canaliculata is very high in number. Although maculata present with the species but very low in number. We do not know why this is happening and why this species is high and low. Is there any specific competition between them? we don't have an answer. I will try to find out the answer later. So later I went to understand the phylogeographic dynamics and the population genetics from this. We did some bioinformatics analysis, and we started with the analyzing their phylogenetic relationship, haplotype diversity, nucleotide diversity, as well as their network, and some evolutionary index to understand that what happened, how they are capable of the question I asked that is the genetic diversity responsible for the establishment? Here I want to find the answer. So this is I'm sorry, this is a very big one. This is the whole snail I collected. I made a phylogenetic tree where most of the snails represented by Pomacea canaliculata, and we have few outgroups from the anciovatidavis to support our hypothesis. And then we have three different networks for Pomacea canaliculata. We only have one specific network for Pomacea maculata. So later, when we summarize this one in the Bayesian statistics, also we have found that Pomacea canaliculata generally have three different clades, and this network as I match up with the haplotype network. So Pomacea maculata generally present with a single network and few representative in total. So in this way, with the phylogenetic, we can say that we clearly distinguish and identify based on the DNA barcoding, and after the sequencing, we can identify those species. But the maximum likelihood tree also support this hypothesis, but the less value of the bootstrap value here is suggesting that there might be like something happening between those two species. We need to understand more details. The mitochondrial DNA data cannot like fully differentiate it, though we need to progress more. But with this data, I started to understand the haplotype diversity of the species. So I created a haplotype network map, and we found that the three haplotype network present in Taiwan. This only could be possible when like a three haplotype different kind of network can only present possible if the introduction happened multiple times, not only one time. So the presence of three haplotype networks suggested that these snails came from different places multiple times to Taiwan, might be imported. But the Pomacea maculata only single network present that suggests that the import or the invasion maybe happened one time only. So to understand more details, I went through that. I matched the distribution analysis with the haplotype network of Taiwan. So from there, we have seen that the south part, including the Tainan, Chiayi, the Kaohsiung, Pingtung, have a higher haplotype diversity. And we also have found some unique haplotype from Hualien, Taitung, and Ila. Those are the and in the middle part they are diverse in haplotype, but their unique haplotype is very less. What I'm saying, this table, if we show that the number of unique haplotype that is developing, maybe developed in Taiwan, that the number of unique haplotype in the Hualien and Taitung in east position, they have a 2 or 1. And the number of unique haplotype in case of Chiayi, Kaohsiung, and Pingtung and Tainan, they have some unique haplotype, and as well as Ila. But in case of middle part, they don't have any unique haplotype, they all have a shared haplotype. If we go to the haplotype diversity, we can see that the Taitung population, the Taichung population, as well as Ilan population and Pingtung population are the highly haplotypically diverse population. In case of followed by other population like Chiayi, like Kaohsiung, like Tainan, they are also haplotypic diverse population. The less diversity was found in the Hsinchu and Taipei zone. If we go to the understand there are more details of the diversity, we went through the Thajimasdi and FUFS analysis to understand how population is growing. So the bold value is like significant p-value, suggesting that the plus value of all FUFS and Thajimasdi population here, suggesting that population is growing exponentially here. And the minus value where they suggested that the population might have faced a bottleneck, and then they are stabilizing there. So all those positive value and the successful p-value suggesting that those population are like exponentially growing in Taiwan, and the genetic diversity is responsible for the growth of population. Later we understand the significant difference between the population. Is there any difference between the population? And this is the gene flow, how much gene flowing between the places. So when you try to understand the pairwise difference between population, genetic difference coefficient, and also gene flow, So we have seen that the Taiwan population is like very differentiated from other populations. They have a huge difference from other places. They're a little bit unique. Followed by Changhua and followed by Hualien. They have a unique population. And we also try to understand that how the gene flow is affecting. As Taiwan is like very not a very big separated place, so understanding this factor will be like need more crucial details to know that if there is really and there are other haplotypes. From here we can say that yeah, Taiwan may be the possibly the center of like haplotype distribution, but we need to understand really more. We cannot conclude here. If we understand the whole picture of the world, what happened here? This is the home of the species. And here is the Argentina, it's a huge haplotype diverse places from here. And the Uruguay is nearby country, Chile, and the USA is the invaded zone, it's not a native place. Here is the Southeast Asia. When this snail first time came here in the Taiwan, and then from the trade, they separated in different places. In literature, they have written down that, yeah, they introduced in different places, but no molecular data from Taiwan, we cannot prove this concept. Here you can see that the diversity of haplotype in Taiwan and compared to the diversity of haplotype in China. So what I was saying, that founder effect, if invasive species go into the another places, this is called founder effect. And if this species introduced directly from this place to another place, this is called Brigitte effect. So founder effect generally bring more diverse population than Brigitte effect. So this we can, with these two concepts, we can prove that Taiwan is the center of invasion of the species. So another species, if you consider another species, in the world we have 7 haplotype network of another species. 7 haplotype network, but only in Asia, presence of 1 haplotype network in Asia. So in Asia, the center of haplotype is this one, haplotype 1, and this is represented by Taiwan and shared with Argentina. In literature, they say that the Pomacea maculata, their originated place is Brazil, this one. You can see that huge haplotype diversity in Brazil. And they say that from Brazil, we bring this snail to Taiwan. But in our study, we found that no, not from the Brazil. We like from Taiwan, they brought this snail from Argentina. But this snail originally from Brazil, but previously developed population in Argentina came into the Brazil, came into the Taiwan. If we find out there's direct connection with the Brazil, then we can say that this population is like from Brazil. But this Pomacea population is like native to Brazil, they bring to Argentina, and from Argentina we take all the snails in the group of snails and bring it to Taiwan. This suggests that the network analysis suggests that this hypothesis could be true. And if we go to the details of this, what I'm saying that the Pomacea maculata, Taiwan is the only place which is like highly diverse for haplotype diversity. And others like New Zealand and if we consider the specific island, like in the Asia, Japan and China, they do not have that much haplotype diversity compared to that. So if we go to select the table, I also make the analysis of whole country, the number of sequences present here, and then haplotype diversity. You can say that this is the native places where higher haplotype diversity is 0.91, 92 in case of Argentina. And in case of introduced places, 0.53 and 0.54 in the haplotype diversity. The number of haplotype reported from Asian country, the highest one is from Taiwan, is 16, compared to China is 12, and the Philippines is 12, and other places are less than 10. If we go to the another species, Pomacea maculata, here you can also see the same result. The native places had higher haplotype diversity compared to the introduced places. In introduced places, the highest amount of haplotype in Asia is from Taiwan, is 4 haplotype from Taiwan, and other places like 1 or 2 haplotypes reported till now. Now, if we understand the pairwise difference between those snails to understand their event of genetic invasion, as I said that 3 different networks present might be responsible for the multiple introduction. How we prove that from here? The bimodal distribution or multimodal distribution of Pomacea canuculata and maculata possibly suggesting that they also bring, they also came from the multiple different location. As well as if we analyze the world population, they have a multimodal distribution also. This suggested that these species have high introduction history in different continents or different places in the world. Now, what if I tell you these species are hybridized together in Taiwan? So what generally happens is Pomacea, canaliculata and maculata, if the hybrid produce hybrid and they have a hybrid and they can mate with hybrid and then they can make it maculata and all the other things. So we need to check the hybridization. What generally happens if we quickly follow the theory, if there is a female Pomacea maculata, the mitochondria generally come from the female and then the nucleus is divided by from male and female and they have a hybrid here. And we need to detect those hybrids. How can this possible? We can detect the hybrid species from the snail. So we target the nucleus sequence. In the mitochondrial sequence, we cannot detect the hybrid. We need to target the nucleus sequence. We need to find a particular zone that can differentiate nuclear gene that can differentiate these two species. So with this one, if we can differentiate those species based on the nuclear, and if they are really hybridizing, so they will have a different combination. The CC is represented for canaliculata that have a mitochondria of Pomacea canaliculata. Pecan is the canaliculata here, and the nucleus of Pomacea canaliculata. Both. This is the pure one. If the CM is the hybrid, if the mitochondria of Pomacea canaliculata and the nucleus of Pomacea maculata, maybe we will say that this one is the CM type. And if the CB type is like, if the mitochondria of Pomacea canaliculata made with the nucleus of both, if they have a both of nucleus, then we will say CB type. Then simultaneously we also have MM type, where the pure Pomacea maculata, the mitochondria of Pomacea maculata, and nucleus of Pomacea canaliculata. We also have the MC type, where mitochondria of maculata meet with the nucleus of it. We also have MB type. So these are the general 4 types I've shown you to understand this thing more clearly. So how do we differentiate those species? We find out that in the EF1 alpha gene, they have a restriction site. The restriction site is absence in Pomacea canaliculata, but present in Pomacea maculata. So if we digest this one with this restriction enzyme, we will find no product in case of Pomacea canaliculata, but in case of maculata, we will find real product, that is, we will find a 2 different band, 190 and 330 base pair. What happens in case of hybrid? In case of hybrid, we will find 3 different band, in 520, 330 and 90. Let's explore this one with our technique. So this is our one summarized result from where we are getting hybrid bands, and some of we are getting like single band. But how about the 2 band? Is there like any differentiation? So when we analyze all the samples, we find out this one from the whole Taiwan. This is the good sample name from Warrior, Taiwan, Chung, like this. So we found only 4 type of group present in Taiwan: CC, CB, MC, and MD. There are no pure maculata in Taiwan. There are currently no pure maculata in Taiwan based on the genetic evidence. And in Asia also, we also find out that in Asia there is no pure maculata present, except only one reported from Hong Kong recently in 2021. So why this Pomacea maculata number is also low and their pure bit also low? What can be the result of this one, or what can be the main cause of this one? So when we divided this one, in this part we have like in the south part we also, as we see the genetic diversity, and we are also seeing that the number of hybrid species and kind of hybrid was also more in case of south part than other parts. In case of Taichung and Taoyuan, we find in hybrid species all. If we divide it based on the 3 part, east, middle, and north, we also detected the south part, the more hybrid species. I'm going to explain why this happened. So if we clearly summarize this picture, this looks like this one, the kind of distribution of hybrid in different parts in Taiwan. So if we go more precise, now you need to understand if their snail species have a parasite inside or not. If they have a parasite, there is multiple, like double dilemma we need to deal with. So those snail species are generally act as an intermediate host. And in Taiwan, as I wrote it in my literature review, I summarized all the data of the cases that happen in Taiwan, and many cases lead to also death for meningitis in Taiwan. So this snail can, if someone eat this snail directly, they can have the chance of growing infection, and also they can also develop in contaminated food, and might be also possible, as I am suggesting, the food, the water transmission of this disease. This is not happened till now, but might be possible. So we collected those snails and we tried to understand their the presence of parasite. This is the quick protocol I have collected, how I digest and how I see the parasite inside. So I like to skip a little bit fast. We identify its parasite based on the morphological character presented here. And coming to the main thing is like we detected a higher percentage of parasite infection in Taiwan. It's very high. Compared to the other Asian countries, they have detected 40% or near about 30%. In some places, we have detected that 70% of the snails are infected in Taiwan. That's why in case of report, when we studied the report, the time when someone ingested the raw snail, they got infected very soon. The intensity of the parasite is very high there. So that's why if they intake one or few snails and they got infected very soon. In case of overall infection rate, we have detected that the middle part has have like more than like 70% infection, and we are followed by the other part. But all of the part have a over 40% infection rate, and this infection rate, can also vary on the different places and based on this one. So this suggesting that the snails carrying the parasite and might be dangerous for us. So coming to the main thing, if we know that these things together, we understand these all things. How this takes me like 3 years to do these things together. If I say that we can do this thing in 3 months, all the things we can do in 3 months. So this environmental DNA comes to play. So this is a technique. I'll say it's when at the beginning we started struggling with developing this technique for 3 or 4 years. How we detect the DNA from the water and then filter it, and then from there we can detect the biodiversity of those snails, and even we can go beyond the present absence state. So this is started. Environmental DNA is recently picking up and accelerating their studies more. You can see that from 2019 and 2021, how they are growing rapidly. And we started near about here, and we do not have any idea what protocol we should follow and how to do this one. There's a multiple thing need to handle, and we do not have an idea to do it. So we try to standardize our protocol and try to develop this technique and try to fit with the environmental DNA community. So this basically the types of environmental DNA you need to follow before you are sampling even what factor you are targeting. So we are targeting here the extracellular DNA, organelle DNA, as well as cellular DNA, but mostly extracellular DNA. I made a chart of how this technique is developing. I'm very excited to study the history of how the technique is developing from in 2008 first environmental DNA study to 2022. How the factor is like developing. I made this one in my research, PhD research, and I try to understand which factor is like causing. If I try to understand the subject very well, I can think after this one. I don't need to think inside the box. I can think out of the box. So I like to do this thing. From here, I find out that there is no study about the snails and like apple snails on environmental DNA. There's no study about the species interaction. There's no study about the population genetics with environmental DNA. So this we standardize a figure. This is from our lab. We made a standardized figure that means from where we need to use the control to use the environmental DNA, in which stage we need what kind of control to make the data viable and to show that people that our data is like 100%. Our data is correct to show represent with the DNA barcoding. So our study was basically we did the two studies, the DNA barcoding and meta barcoding. We did standardized DNA barcoding in Western as well as we did field study of 41 locations. And we also did the meta barcoding study to study the species interaction in light speed and traditional field. So our goal was like primer development, primer validation, and the choice of proper method. We selected few methods and randomized the choice of method, and large scale detection and regular monitoring, community level monitoring, as well as species interaction. So this was our first method of standardization of primers, where we developed a single primer that can have 171 base pair, and I developed one primer that PC is the canaliculata, PM is the maculata, that can detect these two species separately. So I used the primer one for canaliculata and maculata and row wise like this way. So you can see that the canaliculata primer detect canaliculata but could not detect the maculata primer. And the maculata primer detect there is a canaliculata. And maculata primer only detected by maculata gene only detected by maculata primer, not by canaliculata primer. So this is how we developed a species-specific primer. We also have a list of taxa that are given in the supplementary data to develop this primer in silico and as well as that. Then we set up the aquarium experiment and we repeat this experiment and 3 times in every 3 months. we have 3 different aquarium for the positive snail. Sorry, 5 different aquariums for positive snails. 1 aquarium for control with water and sorry. Can I take 5 minutes, please? Let's take a minute. How?
You are not using the CCU account.
I think you can, yeah, so
Yeah.
that's why I stopped in 1
So
year.
you only have 1 hour
Yes.
invitation.
Yes, you can start from here.
Okay.
Can you click the height?
Okay.
Perfect. Oh, yeah. Okay, perfect.
So from here we went to field. We started collecting the environmental DNA. It's like it's sometimes in the hot season, sometimes the dry season, sometimes the rainy. We started collecting the environmental DNA from both Taiwan. This I would like to say that especially emphasis on this one is the Hualian lifestyle as well, like environment is alive for the snakes, but still they are going there. Oh, so we collected the environmental DNA from drainage and aquatic system, and then we came up in the lab and we started standardizing those techniques. At the beginning, we compared the mesopotam and field experiment with only Chiayi and the Taiwan, and we found that we have a high detection rate of Pomacea, caniculatum, and maculata in mesocosm in aquarium, but not in the field. Field detection was very low. But due to the inhibition, this may happen, and we try to solve the inhibition with the PC inhibitor, and then we also try to put internal amplification control to solve this problem. And later, when we solve this problem, we find out this one. Yeah, we detected Pomacea caniculatum from every location of sampling event, and we detected Pomacea maculata. The detection is low. This can go with the distribution number of Pomacea maculata is really low based on the abundance. But surprisingly, when in the DNA barcoding data, we detected Pomacea maculata from the south part only and from 2 other parts. But with the eDNA data, we detected the Pomacea maculata in almost every sampling event except Taipei, Hsinchu, and Miori. The 4 part, we detected from the 11 part. So we have additional biodiversity information with the eDNA data that detected the Pomacea caniculatum and maculata from the different part of the Taiwan. So here we can see that eDNA barcoding generally overcoming the information of DNA barcoding that can help to detect more presence of the things. And if we can summarize in the 4 different parts, we detected Pomacea maculata from every part. In case of DNA barcoding, we only detected Pomacea maculata from south part and few other locations. But in case of eDNA barcoding, we detected Pomacea maculata from every part. That means their presence, but very low in number. We could not detect by collecting themselves, and the eDNA data can detect them with the water sample, and that's very interesting. So for going through more, we want to understand what's behind the scene of the community interaction in the rice field, what's happening there. So we collected the sample in 2 growing seasons of rice to understand that how eDNA data work and how long, how many species are interacting with the rice, and is there any effect of invasive snail on native snail on the diversity. So we started collecting this data from traditional field and organic field to check out that if those both fields have the same effect or not. You can say that the number of pesticides using this one here. So later we come out with, we use the metazoan primer, common metazoan primer, and later we come out with this data. We have a lot of taxa that is unidentified as usual, as we detected in the DNA barcoding for eukaryotes. But most of the taxa reads are from arthropod, generally insect that's coming to interact with the rice. And we have also detected some fungi and bacteria. Later I want to screen down and delete everything and extract my target taxa. So we went there to check that which order are more in abundance. In case of arthropoda, we have a high amount of these orders, and we also have some other orders from algae and fungi. So we want to check that their differentiation in diversity. So we found that yeah, the diversity may be affected by the sampling effect. They also have a differentiation in diversity in organic and traditional field and before and after sampling. But this, if we want to prove this one, we need to increase the sampling number. We had only 43 samples. In case of this one, we need to increase 100 samples, over 100 samples to say this one and to prove this one. We also did some PC analysis to understand, this is horrible to understand because this was not my target, but still I'm showing this now. Now coming to the main point here, to understand that what kind of species interacting together. So we also made a one ternary plot to understand that what factor does like make a differentiation between the organic and traditional field. But the story related to this one is come from here. We extracted the 83 metazone ASBs from the our NGS read, and we identified 34 genera, main genera present in that read. And those 34 main genera are represented by some of the positive and negative interacting species with rice. Some of them are rice-based, some of them are helping to grow the rice. So what are they like? Before I summarize them, this picture is from internet. I'm not able to collect all the features together. So you can see that some of these are the rice-based, serious paste of rice. Some of them are like helping to grow the rice and make what is the fertilizer. And there is a bioindicator of what pollution there. And these are the native snail. This one and this one are the native snail. And this one is our Pomacea canadiculata. And this one is like the common insect, common birds that interacting with the rice field and so on. And we also detect some amphibian DNAs like, but most importantly, this one is very dangerous because this is like Japanese encephalitis flu DNA and the mosquito DNA was high abundant in the field. That means that in the stagnant water they are laying egg, and that could like in future can develop flu on those things. We need to control this one from the rice field. This is another thing. And the top 10 abundant taxa, if we detect the top 10 abundant taxa, the species, the invasive species are among them. The top 10 abundant group in our DNA sequence, the invasive species was among them. And there are also some indigenous species, and as I said, the mosquito, they are laying eggs inside the field, and that could be dangerous for like developing disease. So these are the heat map of those species I found here. I'm not going to very differentiate the traditional organic field here to like I'm going to go smoothly with my hypothesis with the next slide. Yes, this is the native snail and this is the invasive snail. You can clearly see that this color represents the invasive snail and this is the native snail. You can clearly see the clearly dominance of invasive snail over the native snail in every field except this field. So this suggested that the native snails are like dominating things, but I'll try to add one thing from literature. These snails are not herbivorous. These are omnivorous. They also eat native snails. They also eat other insects. So kind of different interaction happening with this native snail. So this also causing threat economically as well as ecologically. So I like to say my main outcome from this present study. So all the study, like we have detected Pomacea maculata in Taiwan for the first time with our sampling event. Before, we do not have any idea this species is present. So this is the first report of this species from Taiwan. So we detected that these two species are living sympatrically in Taiwan. We detected the actual invasion path from in the Asia, how this is invaded. I would like to say that the thing like we have seen the hybrid of this species and the less number of this species. So our hypothesis is like, I will say the previous hypothesis first. Hayes et al. Hypothesis is like Pomacea canadiculata is the dominant species because of the tolerance of the atmosphere in Asia. And this could not tolerate the atmosphere in Asia, and this more like to live in the temperate zone. But our hypothesis is like, those two snails are being together, but might be Pomacea maculata came as a hybrid in Taiwan. This is our own hypothesis, so we try to say from this data. Yeah, the Taiwan was the center of the invasion of Pomacea throughout the Asia, it's true. And the genetic diversity is playing a key role for Pomacea establishment in Taiwan. And if we need to figure out, if we need to mitigate, we need to start from the south part. We need to start mitigating from the south part so that is their population more diverse. If we kill other places, the south part population will take over those places. So the distribution of Pomacea may be responsible for land use, how we use agricultural land different places, and also the human activity. because these species are closely related to and love to reside near the human places. And yeah, these species are hybridizing in Taiwan. That is very bad news because they are like they will try to make themselves establish in this environment and kill all the other native snails and alter the species interaction here. They have a high life-threatening disease, and they may have other also disease that can cause. We can need to see, and we need to like figure out this situation very quickly. And now, if we do mitigation, if we clear those snails very rapidly, we need a strong monitoring method to detect their presence in the environment or not. We cannot pick out that they will bury it in the soil, they will go inside. We cannot pick up that. Here, our newly developed environmental DNA method can help the government to rapidly monitoring or regular monitoring in each and every year in a very short period of time. We can also monitor in the same sampling event the species interaction happening with the Pomacea species. So with this all study, we like to say that with this data suggesting that this species is very dangerous for Taiwan, and I would like to we would like to draw the attention of government to make a strong policy to remove the species and use environmental DNA technique to monitor them, and they can be like have a strong mitigation feature. And then in the future, we need to study about the species interaction, more details we can do with DNA technique. And now recently in 2021, we have, 2022, sorry, we have standardized a methodology to detect population genetic data from eDNA. And we can also implement in the next stage of this project. So here I have like published few papers on this topic of the trend, and some of the paper I have submitted in the journals, and some of them are like in progress for sending in the different journal things. And with this, I would like to thank my supervisor, Chiayi, and Cheng, and Professor and especially the Christian Daniel also. He helped me a lot in this. And I would also like to thank the Captain Stewart from Netherlands and Professor Doi from Japan. We have a strong collaboration to work together in this field of study. And especially this happened due to my professor. The professor is like very kind to me. He like let me to do everything by myself, and I just reached them, and then we just collaborated and learned a lot during my PhD study. So this is my lab group. I'm very helpful from the lab group and all the friends that I have here. I especially like to thank the CCU because this gave me a very good atmosphere to study my things. So the last 4 years was not easy, but not very bad. I'm very happy to finish these things